Hi, my name is Gamrasni, and I'm a Postharvest researcher in MIGAL Institute. The topic of my presentation is calcium treatment of apples increases lenticel breakdown and alters cuticle structure. This work is a collaboration between my lab and also Chaya Friedman's lab in the ARO in Volcani Center. Most of this research was done by the students Pragna and Vikram. Okay, so apples are supplied with all year round, and they are stored in controlled atmospheric conditions at low temperature. Physiological changes occurs during the storage, including a softening of the fruit, but also there are developments of disorders like beta pit and lenticel breakdown. Lenticel and beta pit and softening are common in storage and are related to low calcium level in the fruit. However, the cause for lenticel breakdown is still not fully understood. The lenticels appear as small spots on the apple peel at harvest. However, they might become dark and increase, they become bigger and sink. And this might affect the economical value of the fruit. To follow this damage, we developed a lenticel breakdown index as described here. To enhance lenticel breakdown, we dipped the apples in calcium solutions. And we see that there are varieties which are more sensitive to the lenticel breakdown damage, like Gala apple and Red Delicious, and less sensitive like Granny Smith apples. In addition, there was difference between the years. For example, with Red Delicious apples, at 2014, there was more damage compared to 2015. We used microscopic analysis of the peel to see, and we noticed that the wax and the cutin are reduced, the thickness is reduced due to the calcium dips. And in addition, looking at the surface of the peel, we could notice that in the calcium-treated apples, there are less lumps compared to the untreated fruits. Those results led us to suggest that the reduction in cuticle thickness can contribute to enhanced lenticel breakdown. As you know, the pre-harvest calcium application serve to prevent beta pit development during storage. So what we did in the experiment, we conducted an experiment in the orchard with calcium sprays to see if there is any effect on this damage. We sprayed calcium, calcium chloride, calcium nitrate, or potassium chloride in 2 orchards, Matityahu or Fichman. Those are located at two different climates. And after the total of the sprays were 8 times every second week. Then we sampled the apples from separately from 2 sides of the trees from the north and from the south. And then we stored the apples for 3 months at 0 Celsius. And after that, we measured the lenticel breakdown damage. As expected, lenticel breakdown was increased after storage in the calcium chloride treatment, and you can see that it is more at Fichman orchard compared to Matityahu, and especially it was in the northern side of the Fichman orchard. Looking at the temperatures that were monitored during the development of the fruit, we noticed that at the last stage of fruit development, the temperature in Fichman orchards were much higher compared to Matityahu. And this might have affected the severity of the damage. Okay, so in order to examine the post-harvest effect of calcium, we continue with post-harvest dipping of the apples in calcium solutions. We harvested apples, different varieties, at the ripening, at the commercial ripening stage, and treated them with calcium chloride, calcium nitrate, potassium chloride, magnesium chloride, or they were exposed to 38 Celsius. 4 days after the treatment, we measured the peel strength, the cuticle thickness, and also analyzed the transcriptome. This was 4 days after the treatment, meaning that this is the short-term effect of the treatments. More apples were stored at 0 Celsius, and after 3 months, we repeated those measurements, but in addition, we evaluated the lenticel breakdown damage, and this was the long-term effect of the calcium treatment. What we found after storage is that that calcium dipping had affected the lenticel breakdown damage severely, but also magnesium chloride treatment had similar effect, meaning that this damage is not specific to the calcium treatment. We used a Sudan 4 staining to measure the thickness of the cuticle. This in here in black is the wax layer, and orange is the cutin layer. We measured that together, it's the cuticle, and we noticed that the thickness of the cuticle reduced by the calcium dipping at different varieties of apples. This was at the short term, 4 days after the treatment. In addition, we measured the strength of the peel, and we could see that the calcium treatment and also magnesium treatment reduced the strength of the peel during storage in the sensitive varieties. We don't see it in the untreated fruit, in the control. Using a microscopic analysis, it was indicated that magnesium chloride treatment affected the appearance of the peel structure, and all differently than the calcium chloride treatment, both different from the control. And this is at the short term, 4 days after the treatment. And we could see this effect on the fruit peel also after storage, which is a long-term effect. The peel appearance is different than the control. Okay, so to deepen our understanding on the effect of calcium on lenticel breakdown, we analyzed this transcriptome of the apple peel. We did that from the peels of gala apples after harvest to learn on the short-term effect of the treatment, and after storage to learn on the long-term effect. In addition, we did that from a non-sensitive variety to lenticel breakdown, the pink lady apples, and the different treatments that we extracted the RNA from were control, of course, calcium-treated apples, magnesium chloride, and heat-treated. The PCA analysis divided between samples of apple varieties and sampling time, but not between the treatments. Okay, so when we used Venn diagram, we identified a common and specific genes that were upregulated or downregulated between treatments. And we could see that calcium affected especially the genes by downregulating those more than the upregulation of genes, downregulate. Okay, and KEGG analysis helped us to identify the specific metabolic pathways that were affected by calcium treatment. And it was found that calcium specifically affected the cutin, the suberin, and the wax biosynthesis pathway genes, and also the zeatin pathway biosynthesis pathway genes in gala apples, but not in pink lady apples. It could be noted that calcium had affected and reduced the expression of those genes at the short term here, and also at the long term after storage, a reduction in the gene expression that we did not observe in the pink lady apples. This figure summarizes the work of a few years. Here we can see the biosynthesis pathway of, in purple, it's the cutin. In a light blue color, it's the wax. And in white, it is the area where our genes for suberin biosynthesis. In red color, there are genes that their expression was reduced by calcium after storage, and we already had reported on their reduction of expression. Here we add the long-term effect of calcium treatment on genes and short-term effect of calcium on genes, which were mostly related to the suberin biosynthesis pathway. To summarize, a post-harvest calcium application reduced the cuticle thickness, modified its structure, reduced the cuticle-related gene expression, and reduced the peel strength. All together can contribute to increase lenticel breakdown damage. Calcium had a short and long-term effect depending on the season. We noticed that magnesium also increased lenticel breakdown, but probably by a different mechanism than calcium. The calcium effect occurs also due to orchard calcium application. However, it is possibly environmentally dependent. We would like to thank the apple growers from Matityahu Orchard, Yuval Ogni, and also from Fichman Orchard, Jamil, and the teams that did this research, the Postharvest Research Innovation Center in MIGAL, and Chaya Friedman's team from the ARO. This research was funded by the BARD, which is a binational agriculture research and development fund. And I would like to thank you for your listening. We will be happy to take your questions. Thank you very much.